Alan Gray here with the Microbiome First Summit, underwritten by the World Asthma Foundation. We're pleased to introduce our next speaker, Dr. Paul Turner. Dr. Turner is the Rachel Carson Professor of Ecology and Evolutionary Biology at Yale University and microbiology faculty member at the Yale School of Medicine in New Haven, Connecticut. Dr. Turner studies the evolutionary genetics of viruses, particularly phages that infect bacterial pathogens and RNA viruses. He also researches the use of phages to treat antibiotic-resistant bacterial diseases. Phage therapy, long overshadowed by chemical antibiotics, is seeing renewed interest since the rise in frequency of multi-drug-resistant bacterial infections. Today, Dr. Turner presents his session, New Yale Center to Advance Phage Research, Understanding, Treatments, Training, Education. Dr. Turner, over to you.
I would like to thank the World Microbiome Summit for this opportunity to present on our new Phage Therapy Center at Yale University, and to also describe some of the biological discoveries that we've made along the way that convinced us that we should move forward with phage therapy as a technology that could help patients. So the three images here are giving away the punchline to my talk. Phages are bacteria-specific viruses that offer one possible alternative for treating bacterial infections. On the left, we have an image that relates to the classic ability for scientists to go out into nature and to look for these viruses in natural samples. I'll describe how that's done. And in the middle is a reminder that we can now characterize these in the laboratory, understand the biology of these viruses, and determine whether they should have usefulness in the clinic. On the right is what's happening immediately, and what we expect can happen further is that phages can be used in emergency therapy when antibiotics fail. Today, I'll mostly focus on pulmonary infections and the ability for phages to treat humans, to be used in treating humans when antibiotics can't do the job. First, let me disclose that I'm a founder of Felix Biotechnology. It's a company that seeks to commercially develop phages for therapy. One way to describe the urgency for this kind of technology is to remind that the antibiotic resistance crisis is here. This is the observation that an increasing proportion of bacteria are resistant to commonly used antibiotics that have been approved for therapy. Now, this is a global problem. It's not as if only certain countries are feeling the effects of this issue. Instead, you see all over the world that these bacteria are on the rise. And worse yet, if you project out to several decades from now, it is expected that it can only get worse, and that annually you'll have lots of human deaths associated with antimicrobial resistant infections. Historically, we have looked to pharmaceutical companies to develop new antibiotics, to discover them and bring them to market. And unfortunately, this is now entering what's called a discovery void. And that's not surprising. They have to get a return on investment if these companies try to develop these antibiotics. And instead, what happens is that they are approved, and then they sit on a shelf as what's called a last resort drug. So it doesn't return the investment. And instead, we have to look to other technologies to step in and try to help as we move forward in time, and more of these antibiotics are going to be failing to control bacterial infections. Indeed, in lung infections, antibiotic resistance is an increasing problem. I'll only highlight here one particular pathogen, Pseudomonas aeruginosa, which is listed by the World Health Organization as a priority pathogen. And that's because it has resistance mechanisms, especially efflux pumps, that provide widespread resistance to lots of antibiotics drawn from different drug classes, and therefore multi-drug resistant strains of these bacteria are on the rise. People can enter the hospital, acquire an infection, and unfortunately face a very high mortality rate. And individuals who have a particular lung structure are very vulnerable for these types of bacteria coming in and causing a chronic infection, where these infections are increasingly resistant to chemical antibiotic drugs. Here I'm speaking of people who have cystic fibrosis a genetic disease that causes widened airways, overproduction of mucus, among all sorts of other phenotypic traits. But in the lungs, what this means is that this is creating an environment where bacteria can come in, cause an infection, it becomes chronic, and it takes hold. The same essentially is seen in those who have bronchiectasis, who have chronic obstructive pulmonary disease. And in the USA alone, you can see the impact here of Pseudomonas aeruginosa infections, and we must find alternatives to antibiotics as we move forward in time. I want to back up a little bit and explain what a bacteriophage, or a phage for short. What is this? This is a virus that kills only susceptible bacteria. For phage therapy, usually the focus is on what's called lytic phages. And in this reproductive cycle on the lower left, what's happening here is that these viruses are attaching to susceptible cells, entering, taking over the metabolism of these cells, turning it into a virus production factory, if you will, and then bursting the cell open through an enzyme that is coded for by the virus. And what happens next is that these particles are released from the cell as it dies, and they can go on and infect cells of similar type. This is a very efficient way for these virus particles to kill cells. And in a moment, I'll remind that that's one of the benefits, one of the obvious benefits of phage therapy. It's essentially a self-amplifying drug. On the right is just one image here of lytic phages that are attaching to the outside of a bacterial cell for which one had entered some time before that and taken over the machinery of that cell to turn it into a virus production factory. You can see those particles being formed inside of the cell. So indeed, these are possible alternatives to chemical antibiotic drugs, and I'll get into that more in a moment. What I want to mention is that some of the early discoverers of phages, especially Felix Durell, back in 1917, he was thinking of the usefulness of phages to kill infections in humans and in animals. And it turns out he was very involved in the early days of the foundation of the Eliava Institute, which still operates in the country of Georgia, formerly part of the USSR, and it's located in the city of Tbilisi. So essentially, this early discoverer of phages, this was long before antibiotics were accidentally discovered by Fleming. And it meant that very early on, he was seeing that phages were possibly useful as a way to kill bacterial infections. It turned out that Felix Durell happened to have one and only one academic appointment in his career, and it happened to be at my institution, Yale University. So for this reason, we named our company Felix Biotechnology in his honor. Let's look at the benefits and the costs of phage therapy. So the benefits, I mentioned these earlier. There's a specificity of the phage for its target. And that's quite attractive. It means that you can have a drug that is very specific to going after its target. And it can self-amplify. So it's a drug that makes copies of itself. But you can also see some downsides or some possible costs to phage therapy. These are pretty easily identifiable. One is that if you have too much specificity, this limits the spectrum of the usefulness of a drug, and that's not such a good thing. The delivery to the infection site could be non-trivial. Today, I'll talk mostly about pulmonary infections and the ability for patients to inhale phages, and they travel directly to the site of the infection in the lung. Whereas in other circumstances, other target organs are less easy to reach. And it could be that if you want to target the infections in the gastrointestinal system or elsewhere, you might have to use a different and more elaborate way of getting to the site of the infection. Immune recognition is an enduring question, and it's uncertain whether some phages would be easily recognized by the human immune system and the production of antibodies that would see them as foreign invaders and attack them, limiting the ability of this technology to work. As an evolutionary biologist, there's an obvious cost of phage therapy that I've studied and I'll focus on in a moment in the coming slides. And that is bacteria are very good at evolving, including their ability to evolve phage resistance through a wide variety of mechanisms. So in essence, you'd have to think about this therapy could be awesomely useful in its potential, but I'd also expect it to fail pretty easily if the bacteria evolve resistance and they'll escape phage attack and therefore stop the therapy from working. And here is a reminder of the very many ways that bacteria can evolve resistance to phage attack. On the left, it's a description of what are so-called innate defense mechanisms, changes at the cell surface of the bacteria, such as protein binding sites that the phages would ordinarily use to infect that cell. If those evolve through time and change, then the phage would not be able to recognize that cell as a target. But even if the nucleic acid enters the cell and the phage initiates the infection, it could be after the fact that it is recognized by restriction enzymes and cut up and destroyed before it's able to take hold of the molecular machinery in the cell to make phage copies. On the right, it's a reminder of some more elaborate and more recently discovered mechanisms, especially CRISPRs, where here, in essence, the bacteria recognize phages that tend to attack the bacterial population, build up an array of genetic recognition sites so that immediately, if those phages enter the cell, these mechanisms are deployed to stop those phages from replicating. And it's kind of surprising that this took a long time for microbiologists to figure out that this was happening in some bacteria, but indeed it's something that's a bit akin to adaptive immunity in hosts like humans. So there's a lot here that stands in the way of phages being potentially useful in killing bacterial cells because the bacteria can evolve resistance against them. What are you going to do about this? Are you going to wring your hands and say that this is an impossible technology to move forward? Well, maybe not. What we've been studying is that evolution is a very potent force in biology, but it often involves compromises, and these are called trade-offs. So in very many biological systems, it's easy to see that natural selection can indeed improve traits, but it often does so at the expense of reduced performance in other traits. I always like to tell audiences an example from humans as a way to just illustrate how even in the human species this can happen. So when you consider what humans do better than any other animal, arguably it's our ability to hurl objects with accuracy and speed that sets us apart from many other organisms in the animal world. And the best of us that do this do it professionally, as baseball players, for example, or as softball players. Why can we do this? It's because we have an upper body musculature that comes along with being different than other great apes, in the sense that we're the only great ape species that walks around. We've evolved to walk around on 2 feet all the time. And it frees up our upper body musculature to evolve in a way to do tasks that other animals cannot do, even our close relatives. Now, the problem is that this has liabilities. It puts a lot of strain on your neck, your shoulders, and your lower back to be walking around on 2 feet all of the time relative to our close relatives that do not do this. So automatically, there's a liability to bipedalism in humans, and it leaves us very vulnerable to back problems and neck problems, etc. So it's a reminder that no species is the pinnacle of evolution. All species have traits that have some liabilities associated with them. Let's think about this concept and how we can examine phage therapy, maybe modify it in a way so we can capitalize on trade-offs and make this therapy more durable through time. So we call this a renewed approach to phage therapy, understanding that selection has this ability to improve traits at reduced performance of other traits. Our innovation was to find lytic phages that naturally bind to mechanisms that are important in the target bacteria that are responsible for virulence factors or antibiotic resistance mechanisms. And our prediction was that if these phages kill the bacteria in the classic sense of phage therapy, then they will also select for the bacteria to evolve resistance in a particular way. It's highly likely that the bacteria would evolve resistance by modifying those structures and making them perhaps less resistant or resensitized to antibiotics, and to make them less virulent, depending on the phage. So let's look at a couple of hypothetical examples. If you consider this oval here in the cartoon, that's a bacterial cell. And what I'm showing is an illustration of efflux pumps in some bacteria, including Pseudomonas aeruginosa. These protein complexes are responsible for pumping antibiotics out if they manage to get inside the cell. And this can work across very different drug classes, providing broad resistance to antibiotics, which is obviously a very bad thing for trying to treat these bacteria. So what we conceived of is that if you find a phage that naturally infects these bacteria by binding to the outermost protein of efflux pumps, then in a classic sense, the phages will enter and kill the bacteria. And they'll also select for the bacteria to change those protein structures and perhaps modify them or get rid of them altogether. And what will happen then is that the bacteria become less resistant or completely resensitized to antibiotics. So now you've got a two-edged sword. You've not only killed the bacteria in the classic sense, but you're anticipating the evolution of phage resistance in the bacteria, and you're capitalizing on this by creating a leftover bacterial population that is more sensitized to antibiotics that it used to be resistant to. A faster example would be considering a purple shield around that bacterial cell or a capsule. Some bacteria have this as a way to hide from the detection of your immune system cells. Well, what can happen hypothetically if you find a phage that is specific to binding to the bacteria by attaching to the capsule? Again, they can kill the bacteria in the classic sense, but they'll also exert selection for those bacteria to change in a particular way by ditching the capsule and therefore becoming more seen in terms of the immune system, and they are converted to lower virulence. So I'll give you some examples of this that we've described in the literature and also some unpublished work, but that's the general idea, is that you can have a renewed approach to phage therapy in this way. So I'll take you through some slides that illustrate our discovery pipeline for how we find such phages, characterize them, and become convinced that they might be useful in the clinic. The first is to state something that is very well known, and that is it's very easy to go out into nature and find phage candidates because it's very easy to discover completely new phages in natural samples. You can look in pristine environments like a freshwater lake in the upper left. You can also look in heavily contaminated or human-altered environments like the coastline of Haiti there in the left, or the Democratic Republic of Congo, a river running through it. So with colleagues in those countries, we've discovered phages that are perhaps very useful against some endemic disease problems, such as cholera. But here in the United States, we've also looked in wastewater samples in agricultural animal farms to look for phages that are thriving in association with human-associated bacteria, or on these agricultural animal farms associated with antibiotic-resistant bacteria, because a reminder that antibiotics are used a lot in animal feed to make animals larger and more plump and better for harvesting for meat. So essentially, phage hunting can be done almost anywhere. It's pretty easy and affordable. What you want to do next, in our opinion, is you want to understand what you're working with by doing whole genome sequencing. And we really feel like this is crucial. You can, first of all, look for genes that are specific to lytic phage life cycles, so you can convince yourself pretty easily that these phages that are potential candidates will be killers of bacteria, and not having some more complicated life cycles that I didn't describe earlier, where they only sometimes kill the bacteria and in other circumstances are inherited along with bacteria as they divide and grow. So this kind of a busy cartoon is a reminder for me to say that for Pseudomonas aeruginosa alone, here what we found is a sample of about 100 phages coming straight from nature. And through whole genome sequencing, we've placed them into known virus families. We know what their relatedness is to one another, and we can look exclusively and explicitly for the kinds of genes that should be perhaps avoided because they might carry toxin genes or other things that would be dangerous for the patient in the clinic, and we would like to avoid those. So essentially, you can do a lot of phage genome sequencing to know what you're working with these naturally sampled phages. Now, host range. The overwhelming popular idea is that a lot of phages may be too specific in terms of their killing of bacteria, that if they're not killing enough strains or genotypes of the target bacteria, then they simply won't be useful for a clinic. Now, I think that that is a popular idea that unfortunately dates back to some well-studied phages of E. coli from the last century, in the assumption that what was observed there should be happening for all phages, and that's just not true. An example of why this is not true is on the left. What we've got here is a large library of candidate lytic phages lined up along the side of that rectangle, and across the top are clinical strains of Pseudomonas aeruginosa. So in this grid, everywhere that you see a dark blue square, that's where the phage on the left was able to kill the challenge strain across the top. And you see a lot of dark blue in that grid. It means that indeed a lot of phages coming straight from nature have a very broad host range, suggesting that they would be good as generalized killers of some target bacterium, and you wouldn't have to worry about them being so specific that they would not be useful. On the lower right is kind of a different way of looking at this. You can, in this case, it's a smaller data set, but again, here the challenge hosts are on the left and the phages are along the bottom. But what we're showing here is not only a square indicating when it's filled in that the phage was able to kill the bacteria, but we're now measuring how productive is that phage. How many particles does it produce each time it infects a bacterial cell? And perhaps you want to focus on candidates where these phages are very good at growing well on the target bacteria, and therefore they might be especially good for use in the clinic. Now, a last thing to say on this busy slide is that these data can be pretty complicated to make sense of, but in a future world, if it's easier and easier for a physician to know what the genotype is or the genome of the target bacteria that are causing an infection, maybe through that information alone, one could take it to a phage library and choose from the phage library which should be the best phage in killing that particular strain. And this takes sophisticated approaches like machine learning and artificial intelligence. And certainly it's a goal of many companies to try and develop this kind of technology to make it more efficient in terms of predicting exactly the match between a phage and its possible target strain. Okay, so far I've said phages are easy to find in nature. You can sequence them, and you can easily see that some of these phages are good at killing a broad spectrum of strains of the target bacteria. But really, for our trade-off approach to work, we have to know what the phages are binding to. We would have to know that the virulence mechanism or the antibiotic resistance mechanism is not only used as the binding target, but can be predicted accurately that if the bacteria evolve away from phage attack and change that target, that something good would come from that biomedically, like conversion to resensitivity to antibiotics or a reduction in virulence. And I'll give you examples of that. But first, I want to show you a high throughput method that we developed as a way to try and figure out what phages are binding to, especially if they're newly discovered ones that you want to consider moving forward in your pipeline to make a claim that they would be good candidates in the clinic. So what we did here was take an existing technology of transposon insertion, or INSEQ technology, where you can take a library of bacteria and use transposons that insert at individual sites in the bacterial genome, knocking out the function of the individual genes. And then you expose that library to a phage, and you see what grows out or what's enriched. And in the cartoon on the lower left, we're looking for information that shows up in the blue region. These would be bacteria that are enriched as knockouts because whatever gene they have, it's useful for the phage, and therefore the phage can't grow as well, and the bacteria are protected from phage attack. A very useful control was to try this technology with a well-known phage called T6 that infects E. coli, and has been known for a very long time to do so by binding to the TSX porin of these bacteria. So this control showed that indeed the highest statistically significant hit in the screen was the TSX porin, shown there circled in blue, and that confirmed something that we already knew. But then we took the technology to a novel phage, one called U115, and it turns out that here again, we find that the phage is attaching to the TSX porin. But the point is that we knew very little about that phage before we did the screen. We knew it was lytic and that it killed E. coli. And this high throughput approach allows us to immediately infer what that phage is binding to and whether or not it could be a good candidate for therapy. So these sorts of high throughput approaches are very attractive because you want to quickly be able to take a newly discovered phage and figure out this detail of its biology. So now let me give you some examples of phages that have been useful, and we've used them in the clinic. And this first one is a phage that targets Pseudomonas aeruginosa, and it's abbreviated as TIVP-H6. That stands for type 4 pilus H6. And we have observed that it reduces the virulence of these bacteria. So it kills them, and then it selects for phage-resistant bacterial strains that are reduced in virulence. And I'll explain why. So what this phage does is it binds to the type 4 pili of the bacteria. So these pili are very important for what's called twitching motility. It's the way that the bacteria can move about, including their ability to move about on cells of the human body and to initiate infection. But type 4 pili also function in many other things, such as biofilm formation, and those are very tight, rigid structures that some bacteria can produce, and it helps them establish chronic infections. So essentially, we've got here a phage that is targeting in its binding something that is very important that we know for virulence in the system. So let's begin with a bit of unpublished data there on the lower left, where we did the screen that I just talked about, and all that stuff in blue are type 4 pilus genes. So now we're very convinced that these bacteria are, if they have these gene knockouts for genes that are used in constructing type 4 pili, that this is causing us to be more convinced that indeed the phage is using type 4 pilus as its binding target. So what's the consequence? If you look in the laboratory and do something called an attachment and entry assay, this is where you can grow cells of human origin, those that are in the airway epithelial layer. And if you grow them in the laboratory and then take the kind of bacteria that are causing infections pre-therapy, and if you take from a human patient these bacteria that are left over after the phages have been used in therapy, those are post-therapy strains. And what we find is that indeed the post-therapy strains of these bacteria are very debilitated for their ability to move across cells and to attach to them. And this makes sense because essentially the binding target are the type 4 pili, and the bacteria are evolving phage resistance or phage escape by altering or removing those type 4 pili. Now, it turns out that this is highly useful because there is a gene network that links up type 4 pilus formation with another thing that we care about in the clinic, and that's piocyanin. This is a virulence factor with toxic effects. It causes cellular inflammation and oxidative stress. And I'll show you here in a moment in this bar graph some convincing evidence that this phage is essentially an anti-inflammatory phage because it kills the bacteria and it selects for phage-resistant mutants that are reduced in their ability to make piocyanin. And that's quite attractive because it's reducing this virulence factor in the leftover bacteria. And that's the trade-off that we were seeking. So what's going on here? In this bar graph, it's a little bit busy, but I'll walk you through it. The blue bars are, if you take certain antibiotics and you take a strain of Pseudomonas aeruginosa and expose them to those antibiotics, many of them could be ineffective in killing the bacteria at all, but they could actually cause those bacteria to make more piocyanin, to be more virulent when exposed to that antibiotic. Clearly, that's not something you would want to see in the clinic if you were trying to use these antibiotics ineffectively. Erythromycin is a very interesting drug because it can calm cellular inflammation, but it wouldn't necessarily kill the target bacteria. So the blue bars are showing you evidence that, for example, on the far left, Cipro is a drug that will cause these wild-type bacterial pathogens to make more piocyanin. Erythromycin, on the other hand, reduces the piocyanin production, but it's not killing the bacteria. So the red bars are better news. On the far right, this is what happens if you take this phage and try to kill the bacteria. Indeed, you do. You're able to do this, but the leftover bacteria are very debilitated for their piocyanin production, and that's why that bar on the far right has such a low value. This works very good in combination with erythromycin and also reverses the effects of Cipro. Whereas the remaining bar there that I didn't describe, that's if you take these phage particles, heat inactivate them so that they're not able to kill the bacteria. And that's a control saying that indeed this effect is due to successful phage infection. On the right is a color image of something that's quite obvious when you use this in the clinic, and that's the wild type pathogenic bacteria. When they make piocyanin in abundant amounts, it has a blue-green color. So when you spin the cells down in the lab, pellet them out, and look at the leftover supernatant, it has this very blue-green color. Whereas what we find is that we can kill these bacteria, and the leftover mutants that are resistant to this phage are completely debilitated for their ability sometimes to make piocyanin. So that's very attractive as a trade-off. And in this case, the leftover supernatant looks pretty much just like the growth medium because the bacteria can't make piocyanin anymore. All right, so that was a quick example of a phage that toggles virulence in target bacteria. Let me now give an example of a phage, a different phage, that toggles antibiotic resistance and converts it to sensitivity. Again, we'll stick with P. aeruginosa as the example, target bacterium, and in this case, it's a phage called OMK01 that we published on a bit ago in 2016. So what does it do? It interacts with multiple receptors on these target bacteria, including the efflux pumps, MEXA-B, XY, as well as type 4 pili. We think it's an interesting phage in its ability to interact with multiple cellular receptors. But often what we see is that in the middle graph, this is the important part, is that the phage-sensitive wild-type bacteria have a particular minimum inhibitory concentration of various drugs that could be used to kill them, and they're very resistant to those drugs. But spontaneous phage-resistant mutants of these bacteria are often showing very different MIC values, where they've become more sensitized to these antibiotics as a way that comes along with as a trade-off to avoid phage attack. On the left there, I forgot to say that that's a structure of an efflux pump in P. aeruginosa, and I wanted to highlight that the upper portion of that cartoon is highlighting the protein that the phage would attach to. So it turns out that this phage is very interesting. It's what's called a jumbo phage. It has a very large genome and has kind of an elaborate way of getting inside of the cell and creating a shielded off way to replicate within the cell so that CRISPR-Cas systems cannot see it. And therefore, these DNA targeting CRISPRs are ineffective against this phage. So I'm telling you this as a reminder that phages have all sorts of interesting biology. And when you use them as candidates and you learn more of their biology, you can sometimes find that they have some tricks in their arsenal. And this is one. In this case, this phage not only causes a trade-off that's attractive for what we're trying to develop, but it also has a way of avoiding many CRISPR defense systems in bacteria, making it even more attractive as a way to be deployed in the human body and try to target the bacteria. And we wouldn't have to worry so much about CRISPR defense systems in preventing them from killing the bacteria. All right, I'll give one more example before I move on to some clinical data. And in this case, again, we have a phage that targets efflux pumps, and it's one that we've characterized and done a lot of work on, especially a paper in 2020, but it happens to infect E. coli. So in this case, we're learning a lot from it as more of a model phage to understand how phages interact with efflux pumps. And again, it is selecting for antibiotic resensitivity in these bacteria. It binds to the outermost protein of these efflux pumps called TOL-C. That's what the related efflux pumps in E. coli are. These are TOL-C efflux pumps. And when the phage binds to efflux pumps, it drives selection for tetracycline resensitivity. So this is seen in the data on the left, where if you look at wild type E. coli, a TOL-C knockout and a spontaneous phage-resistant mutant, and you grow them in the absence of tetracycline, they grow equally. That's what these bacterial dilutions show us when you're observing how many bacteria can grow in that environment. And the data on the right is if you grow them at a particular level of tetracycline, as expected, the TOL-C knockout bacteria can't grow very well because they can't move the tetracycline out of the cell if it gets in. But what you see is the exact same phenotype in the spontaneous phage-resistant strain. And then this mutant, it's debilitated for its ability to move tetracycline out of the cell. And presumably that's because it has gained phage resistance by altering that ability, and it has now become resensitized to tetracycline. So for this phage, I always like to say the plot thickens because it has a co-receptor, and that is LPS, which is an abbreviation for lipopolysaccharide. This is very abundant on the outer portion of bacteria, and what you find is often that phages will come in and they'll bind to some mid portion of LPS as a receptor or co-receptor to initiate phage infection. So what I'm showing you is a side view of a structure of LPS there on the right, and the colored rectangles are the portions of LPS where we know that this phage is binding to. And the consequence is the graph on the right. So if you look at bacteria that are wild type or have certain genes knocked out in this portion of LPS, knocking out these genes that pretty much make LPS more shallow or shorter in length, this is easier for antibiotics to enter, including colistin. So what you find is that these knockout strains, these controls of bacteria, are more sensitive to colistin. And what you'll see on the far right in that graph is all of these phage-resistant mutants show that same phenotype, is that they've now gained resistance to phage attack, but the trade-off is that they become sensitized to colistin. So we've studied this quite a bit in vitro and laboratory studies, and I'll just summarize briefly something that you can go read in the literature if you have interest. But we isolated 20 phage-resistant mutants of E. coli, and it was a perfect predictor that 6 of these had TOL-C changes, and the consequence is that they became more sensitized to tetracycline. Whereas the remaining 14, as a way to gain phage resistance, they changed LPS and they became more sensitized to colistin. So this was a very perfect prediction of what you call a pleiotropic consequence, that these trade-offs were due to phage resistance in the predicted way. But some bad news is that if you look in the graph on the left, that bar on the far right, these are some of the LPS mutants that are gaining resistance to the phage, and it turns out that they're actually more resistant to tetracycline. Fortunately, there was a trade-off within a trade-off that we showed in the study, and that is the more resistant that these bacteria could become to the phage through one mechanism and changed their sensitivity to colistin versus to tetracycline, it turns out that this helped in the sense of making them more sensitive to the opposite drug. So in other words, there is a possibility for these bacteria to find different mutations, different routes to phage resistance. And even if they gain resistance to the phage and it helps them to maintain resistance to some antibiotic, it turns out in the presence of the other antibiotic, they are becoming even more sensitized to it. So this is an example of a very complicated system where we've worked out the details and we've convinced ourselves that there are trade-offs and indeed trade-offs within trade-offs that could be useful for this moving forward. One last question that we examined in the study is, the problem is you can't accurately predict which of these mutations would actually happen for the bacteria in the infected patient, because that is a completely spontaneous thing. So if you deploy the phage and you rely on mutations of a particular type, that's not possible because the bacteria could change in many ways. What we wanted to study next is that, well, if they could change by altering TOL-C or change by altering LPS, which of those are going to be most likely to occur during the therapy? It helps us refine our prediction. And what we found is that on average, the TOL-C mutants were less debilitated in their growth. In other words, they maintained fitness that was similar to the wild type bacteria, as shown in this graph. So the solid black line, this is over a 24-hour time span. What is the growth of the wild type bacteria. What's its increase in what's called optical density 600? That's just a way of a proxy for measuring bacterial growth. And in purple, we found that the TOL-C mutants grow pretty much equally to these wild type bacteria, whereas the LPS mutants are very debilitated in their growth. And that would suggest that if you deployed the phage and these spontaneous mutations happened in a patient, it's highly unlikely that the LPS mutants are going to be the way that the problem is solved, because they're not very good at growing. And if they could instead have a mutation that alters TOL-C without altering their fitness or their competitive growth, then I would expect that those would be the ones that would win. And we could more refine our prediction to say that TOL-C would be the changes that would be important. So indeed, we did some studies in the laboratory where we let the bacteria grow for 10 days of serial transfer evolution. And we tracked, first here shown in the orange lines, that through time, if there's no phages in the environment, you just evolve the bacteria, you can see that their resistance to tetracycline, it sort of bounces up and down. It's an unselected marker, but it's pretty much maintaining wild type levels. But if you co-evolve these bacteria with the phage, as shown in these greenish lines here, these are lineages of the bacteria that are co-evolving with the phage. And what you see is that they're drifting down in their resistance to tetracycline. And that's because they are indeed resisting phage attack by changing TOL-C, and this alters their resistance to tetracycline, making them more sensitive to it. So this confirms that the growth on the left is recapitulated in these experimental microcosms in the laboratory to suggest that the TOL-C mutations are going to be the ones that are favored. Last, before I go to clinical evidence, would be what we don't want to see is if you deploy a phage, that a trade up is happening. In other words, if you pick a phage and mistakenly use it to kill bacteria, and if the way the bacteria solve the phage attack by gaining resistance to them makes them more resistant to some other antibiotic, then you're essentially just making the problem worse. And we showed in a paper in 2021 that, for some phages, this is the case. We took an example of two phages, T6 and U115. I talked about them earlier. Excuse me. And what we observed is that an antibiotic called albacidin is able to enter the bacteria in exactly the same way as these phages through the TSX porin, as I talked about earlier. And in this case, all the resistance mutations in the system trace to the TSX porin gene, as shown here. There are very many mutations that'll happen. And they all provide perfect resistance to the phage. Okay, so now I'd like to go on to describe some clinical evidence where we've deployed phages in emergency treatment and show you the outcomes. Our first had nothing to do with a pulmonary problem. Instead, this was a man who had an aortic arch replacement. And this is an artificial part of his heart that was installed. And unfortunately, what was observed is that he had a chronic infection of Pseudomonas aeruginosa, such that by the time his surgeon figured this out, opened him up to take a look at it, it had already eroded away part of his sternum. So it was actually quite remarkable that this man who was in his 80s was even still alive, because he had a very raging, large infection by multi-drug resistant Pseudomonas aeruginosa. We used the phage called OMK01 that we knew selected against antibiotic resistance and converts the bacteria to sensitivity to antibiotics when they gain phage resistance. And we had US Food and Drug Administration approved treatment of this man in 2016 by deploying the phage in an ordinarily useless antibiotic near the site of the infection. They synergized together in the predicted way, and after a single dose, his infection was cleared. In other words, we deploy the phage, it kills the bacteria in the classic way of phage therapy. It selects for the bacteria that remain as mutants to be enriched for those that have become sensitized to this antibiotic. And the synergy between the phage and the antibiotic was able to clear his infection. And you can see a picture there of this man on the right in his office when we were thanking him for his bravery in undergoing this emergency treatment. And we were very happy for him, and his family was indeed happy that in his later years, he was able to go back to work as an ophthalmologist in New Haven, whereas for very many years he was out of work, he could not work. He was just in and out of the hospital with this problem. So this received some coverage in the news and reviews, and we were just very delighted that this worked. And the question that we had is, why did it work so well? So indeed, we examined this through some theory with Joshua Weitz's group at Georgia Tech, where we were a bit surprised that one dose was enough for this man to recover from a very, very bad infection. So I won't go through the details of this model because it's published in 2020, but it's essentially looking at the interaction between antibiotic and phage and potentially the immune response. So in other words, if you have the synergy between a phage and an antibiotic, and you reduce the infection enough, it could be that the immune system can kick in and do the job that you expect it would by targeting the bacterial population, pushing it down even further, and perhaps even clearing it. So the red arrow is showing you when all three of these are working together synergistically, and the white space in that lower right graph is depicting how the best outcome is when all three are working together, because you get the greatest probability of clearance. So we are continuing to examine this kind of a possibility for the data that come from our patients to see if we can understand a bit better about why the therapy can work so well. The second case I'll show briefly is a young woman whose case is not yet published. We're hoping to publish it soon. In her case, she has cystic fibrosis. She had a pulmonary infection caused by pan-drug resistant Pseudomonas aeruginosa, and she had shown many years of pulmonary decline at the tender young age of 22. Unfortunately, this is often the case in some cystic fibrosis patients, is that they can face at a young age a lung transplant. And in her case, you can see the green data are drifting down across the years. This is a measure of FEV1, which is how much air can she force out of her lungs in 1 second. It's a very popular approach for trying to figure out lung function in CF patients. And the details on the left will convince you that she had very poor lung function by the time she came to our attention, very high density of bacteria evident in her sputum, and worse yet, all those red R's on the left are indicating that drugs that she might have used and her physician might have used to treat these bacteria were ineffective because it was a pan-drug resistant bacterial infection. So what did we do? We had her nebulize in these the phage that would target this infection in her lungs. She inhaled in 3 times a day phages for, I think it was a 9-day period. And this administered therapy, as we continued to track her lung function, it improved almost immediately and it trended upwards. And the best yet is that not only did her lung function improve to avoid a transplant, but if you look at the bacterial density in her lungs, it had been very reduced. And it's very difficult to reduce all the way to clearing entirely within the CF lung. But if you reduce it down to a very low density, then this is good, and you can just keep an eye out for exacerbations or outgrowth. But best yet is that the remaining bacteria, as we predicted from the laboratory research, is now dominated by drug-sensitive strains of these bacteria rather than drug-resistant ones. And this is a perfect recapitulation of what we saw in vitro and in the exceedingly more complex black box of the human body. We were able to accurately predict that in the classic way, the phages would reduce the bacterial infection. And in our innovative way, it was also capable of switching the remaining bacteria to sensitivity. So there are other patients that we've helped that I don't have time to go into all the details. Over 20 of them, mostly pulmonary, have been treated. There on the left is a reminder for me to say that many of them have CF, bronchiectasis, or COPD. You can see the details of many of them in this table. So this is therapy that we would like to bring through compassionate use to lots of patients who need it. Technically, it's personalized therapy, so therefore it can work, tailored to exactly their infection. But that's, I'll talk about in a moment how that's not good enough. We want to try and find a generalized approach as proven through clinical trial. But the point is, for men and women of various ages and different background problems, mostly Pseudomonas aeruginosa is what we've been targeting here, and we've been doing it very effectively. So some of the clinical samples from these patients give us information about them. They can give us sputum samples that through time we can examine, for example, what are the remaining bacteria in sputum samples to try and prove that these are changed in a way that we're predicting. But first, let me highlight that if you look at their lung function improvement, if you take a collection of 9 patients, that's what we have upcoming in a case series paper, they all have CF, they've all been treated with a variety of phages where we're predicting trade-offs should occur. And across the board, we've seen lung function improvement and avoidance of lung transplant. We've also seen bacterial load reduction, which is essentially what we wanted to observe. And in the lower right, in the cases where we've deployed phages that are supposed to be changing the antibiotic resistance of those bacteria to sensitivity, that has been mostly observed. So their inflammatory molecule reduction is often dramatic, and let me describe what that is next. So here we can do longitudinal analyses of CF patient sputum samples, and now we have some detailed examples from patients where we deployed that type 4 pilus targeting phage that I described earlier. And what we did is isolate clones from sputum, grow them up in the laboratory, and measure how much of the piocyanin virulence factor can these bacteria make. And then the graph on the left, you're seeing that multiple strains taken at independent time points, that through time, the piocyanin production and the concentration of it, when you grow these bacteria in the lab, spin down the cells and look at how much piocyanin they make, it's troughing out such that by the end of the therapy, it's indistinguishable from just growth medium that the bacteria are in. So they're really, really debilitated in making piocyanin. And on the right, the consequence is that you can, again, take those cells pre versus post therapy, grow them in the lab on airway epithelial cells, and look at the human-derived cells and their inflammatory markers like IL-8 and IL-6. And you can often observe with great statistical significance that the piocyanin is changing in its production, and along with it, the reduced inflammation is happening for these human-derived cells when they're exposed to the bacteria post-therapy. And that brings me to the end of my talk, where I'll highlight a little bit of the Center for Phage Biology and Therapy at Yale and our mission, and essentially what we want to do going forward. In essence, we want to advance the scientific understanding of phage biology, translate those advancements into the clinic, and disseminate training and education at our university and indeed around the world to try and look closer at this therapy and its potential. So how we do this is through 4 integrated programs that are a pipeline for phage therapy development. That is essentially what I talked about earlier. Now, this is a multi-million-dollar investment in phage biology research, and I have to thank my institution for that. And our mission, as I stated earlier, it's written for you there. And I just wanted to highlight some of our leadership team. The person who's worked with me most closely on this within my lab is Ben Chan, who's our scientific director of the center. And he's a fantastic scientist who's been working for very many years on phage therapy development. Our main medical partner, physician partner, is John Koff, who's a pulmonologist, and he's really been helping us as the leader of our adult CF center at Yale and bringing this therapy to patients who need it. My lab group has been very instrumental, but especially those individuals in our research and support team listed along the bottom, various postdocs, clinical fellows, research assistants, and even visiting fellows. So we're very happy that people are visiting us from around the world to take part in this research as well. So the program 1 is the phage biodiversity and evolution. This is where we are trying very hard in earnest to sample Earth's biodiversity to discover new phages. We're doing this with domestic and international partners, and we want to understand through basic research, phage biology and evolution, as well as educate ourselves and others about how this technology could be further developed. We've done workshops with Phages for Global Health. That's an entity that tries to take this technology to places where it is very affordable and could be used perhaps soon. So therefore, we've done workshops in East and West Africa and portions of Asia. Program 2 is phage biotechnology, where we are trying to do higher throughput development of the therapy, also taking technology like microscopy, whole genomics, leading edge tools to explore the dynamics of these systems in vitro and in vivo, and to take theory to these problems and try to understand what's going on in the patients and in microbiomes so that we can be sure that what we're deploying is safe and effective and that we know the underlying details of it. So therefore, we're partnering with people with very many different disciplinary backgrounds, biophysics, engineering, computer science, etc. And this is part of my work as the director of our Quantitative Biology Institute and our program, graduate program in physics, engineering, and biology. The program 3 is translational research, where indeed we want to confirm that the phages have the ability to broadly kill pathogenic bacteria. We are doing some animal model work, preclinical studies to convince us that this works in vivo. And we want to further develop these phages that are targeting especially pulmonary infections happening in CF and other patients. But there are other targets as well. So I didn't spend much time talking about the problem of sepsis, post-COVID pneumonia, bacterial pneumonia, and other infections that are also multi-drug or pan-drug resistant. And indeed, phage therapy might be able to help. And last would be phage therapy itself. That's our program 4, where we'll continue to do what we're doing currently, FDA-approved compassionate patient treatment. We'll pursue FDA-approved compassionate treatment in other infections as well. And we also want to ensure that this therapy can make it to everyone, regardless of socioeconomic status. But importantly, the endpoint goal is always to conduct clinical trials, if possible, to further develop this commercially. And I'll end by mentioning SCiFi, which is our cystic fibrosis bacteriophage study at Yale. This is ongoing right now. And this clinical trial is the first in human phase 1/2 trial for phage therapy in CF. So essentially the way this works is we have CF healthy volunteers who are inhaling in phages over a multi-month period, and we are monitoring them through time to see that these phages are effective at reducing bacterial loads in the CF lung compared to individuals who are volunteering but are taking a placebo. And no one is prevented from access to the experimental therapy, so they cross over to the other portion of the trial. So in other words, those who are taking inhaled phages eventually get moved to the placebo group and vice versa. So what we hope to see is that the primary endpoint of the ability for the phages to reduce and control bacterial load in the lung is happening, and we can be convinced that this investigator-initiated trial should move forward successfully to doing a commercially focused trial later on. So I'd like to end by acknowledging the very many people in my group and my fantastic collaborators at Yale and around the world who've been helping us do this awesome work. I'm only as good as the people who work with me, and I've had the pleasure of working with some awesome people in my career. Today, I mostly talked about the work of Ben Chan. He's my primary collaborator on everything that I talked to you about today, and he's a research scientist in my group. Our main physician partners are Gail Stanley, a clinical fellow, and John Koff. I have postdocs within my group, like Katie Courtwright, and Alita Burmeister, who have now moved on. She's now moved on to forming her own group at University of Wisconsin-Milwaukee. Same for Lisa Bono, who's now at Texas Tech University. So basically, the people who've worked with me have been great to advance this research, and many of them have now moved on to form their own laboratories. Some colleagues at Yale, like Barbara Kazmersik, have been essential colleagues as experts in Pseudomonas aeruginosa and other target bacteria. And I today talked about some of the work of Joshua Weitz and folks in his group, like Rogelio, who did the theoretical work that I highlighted. So thank you for your attention. I hope that this was an illuminating talk and that you learned a little bit about what we're doing and can keep track of the further work that we're trying to develop in our phage center.